Alright, so today we're going to be talking about reproduction in plants, specifically angiosperms, which would be a flowering plant, and a gymnosperm, which would be a coniferous plant. In both angiosperms and gymnosperms, we have the male section, the female section. The gymnosperm is fairly easy. There's a male cone on one part of the tree, or on one tree, and a female cone on a different tree or different part of the tree. In a angiosperm, it's all contained in the same flower, but we have the pistil, which contains all the female tissues, and then we have the stamen, which contains the male tissues. Now in the gymnosperm, we have the male cone that produces the pollen. The pollen then is released, and the wind carries it to a female cone, where we have these nooks and crannies that the pollen settles into, finds the egg is then fertilized, and that becomes a seed. The female cone eventually opens up, the seeds fall, and we get a new plant. And an angiosperm is a little bit more complicated. The pollen is contained and made on what's called the anther, which is held up by the filament. The anther then, depending on the type of pollen, on the type of plant, can either directly transport the pollen to the stigma, or if it's a direct deposit that's in a self-pollinating plant. If it is a cross-pollinating plant, the anther needs to somehow transfer this pollen. This is often accomplished with the use of insects, honeybees, moths, butterflies, things like that. So then the anther is transported to the stigma, which is just kind of a sticky landing pad for the pollen. The style holds up the stigma, but then if some pollen gets onto it, the sperm from the pollen then produces a pollen tube, which is going to come down here into the ovary, which contains the ova, which then will be fertilized and become the seed. Now, flowers also have the petals, which attract the insects to it, initially, if it's a cross-pollinating plant, to facilitate that pollination, or later to potentially entice an animal to eat the fruit that is developed as the seed matures, and therefore spread out the seeds. There's also the sepal, which protects the ova and the ovary and all the other structures as the plant is developing and throughout its life. Now, the way that the pollen and ova are created are very different than the way that the petal is created. Petals and the sepal and the stem and the tissues for the filament and the roots all go through a process called mitosis, which creates cells that are genetically identical to the parent cell. In meiosis, they result in cells with half the genetic information, because the male cell, the sperm, will contain half the genetic information, and the ova, the egg, will contain half the genetic information. And then when they come together, then the seed has all of the genetic information necessary to create a new plant. So let's look a little bit more at mitosis and meiosis. So again, mitosis is in most structures. Meiosis is only in sex structures to create sex cells. So in mitosis, we have these different phases. There's prophase, prometaphase, metaphase, anaphase, and telophase. In prophase, the genetic material gets copied and duplicated as perfectly as can be done in the fallen world that we live in. And then in prometaphase and metaphase, the genetic material, the chromatins, line up along the center of the cell, and then these little lines, which are called spindle fibers, grab onto them and start to pull them apart. They separate, go to the opposite sides, and eventually the cell just kind of everything else in the cell just kind of follows. All the different organelles just kind of get divided between the new cells, and we end up with 2 new cells. Now in meiosis, the process is very similar, but there are a couple key differences. In prophase 1, the genetic information is copied, but there is an intentional, say we have one chromatin and another chromatin. They will intentionally swap part of their chromatin to create a new combination of genetics, which is why children don't always look exactly like their parents. Part of it is because half of the information is coming from the father and half of it is coming from the mother, but there's also this slight variation that happens in this prophase where the genetic information gets changed. So we have a different combination of genetics. And this is very useful to prevent things like genetic diseases and inherited issues. Okay, other than that, the first set, you can see we've got prophase 1 and prophase 2. The first set is very similar. So after the genetic information has been copied, it lines up along the middle, it gets grabbed by the spindle fibers, and it separates into 2 daughter cells. That takes us to prophase 2. Now normally in prophase, the genetic information gets copied. In prophase 2, it does not get copied. So say our plant needs 40 chromosomes. So in prophase 1, it would copy and end up with 80 chromosomes, go through, and now in prophase 2, each of these cells now has 40 chromosomes. It goes through, it lines up along the equator, the center, the spindle fibers come, grab it, pull it apart, and now we have 4 daughter cells, but each of them only has 20 chromosomes. And again, because this happens in both the male and female reproductive cells, the male reproductive cells, the sperm, will have 20 chromosomes, the female reproductive cell will have 20, they'll come together, and then you can get a seed that has 40 chromosomes and maintains the correct number of chromosomes and the correct genetic information to create a new plant. So again, in mitosis, that happens in most cells after the egg and the sperm have come together and been fertilized. Then mitosis comes in, and that's what causes the growth. That's mitosis. We start with 1, we end with 2, and they are roughly genetically identical. In meiosis, we start with 1 and we end up with 4. And they each have half the genetic material that has been slightly altered. So we get some of those variations that happen that create a much more interesting universe.